Good morning, good afternoon, and good evening to you all. My name is Antara Sengupta, and on behalf of Bioprotocol Webinars team, I welcome you to our webinar series, New Tools of the Trade in Life Sciences, Technologies that Enable Breakthroughs. For those joining us for the first time, Bioprotocol is an open access, peer-reviewed online journal dedicated to making life science research more transparent and reproducible. We sincerely thank you for joining us from various locations and being part of our mission. I'm excited to welcome you to today's webinar, From Llama to Nanobody, a streamlined workflow for the generation of functionalized VHHs. This webinar is a special one because it's not often that we have the pleasure and honor to have our authors with us as our panelists. Today's webinar covers a promising work which has been published in Bioprotocol's March 20th issue. And so we're delighted to introduce Dr. Lauren Owens as our distinguished speaker and Dr. Raymond Owens as our moderator. Before we begin the webinar, let me share a couple of quick announcements. If you have any question related to the webinar, you can post them in the Q&A and chat sections throughout the talk, and our panelists will address them at the end of the presentation. The webinar will be recorded and it will be posted online on our website as well as YouTube channel. So if you wish to revisit it, you can do so. Additionally, if you would like to receive a certificate of attendance, we encourage you to participate till the end of this live webinar. So without further ado, I'm delighted to welcome our moderator for today's session, Dr. Raymond Owens. Dr. Owens received his PhD in biochemistry from the University of Cambridge, UK, followed by postdoctoral work both in London and Oxford. He then joined Celtech, UK's first biotechnology company, where he focused on antibody engineering and small molecular drug discovery, later becoming the head of new target discovery. In 2002, Dr. Owens moved to the University of Oxford to run Oxford Protein Production Facility, OPPF. He is currently a professor of molecular biology in the Structural Biology Division in the Nuffield Department of Medicine, University of Oxford, and also leads the Nanobody and Protein Production UK, UP-UK projects at the Rosalind Franklin Institute. And with that, Dr. Owens, thank you so much for being here, and I hand it over to you now.
Great, thank you very much for the very kind introduction, and also thank you to Bioprotocol for the opportunity for us, well, Lauren in particular, to present the work that she's been leading in the lab here on the sort of nanobody discovery pipeline. Lauren is an experienced scientist with a lot of background in antibodies, protein production, joined us a few years ago from a postdoc in Poland, but prior to that, a postdoc work in South Africa, which you'll pick up from her accent. And so I think it's a great pleasure to introduce Lauren, and I'll hand over to Lauren now to take the seminar forward. Thank you. Over to you, Lauren.
All right, thanks, Ray. Can you give me a second and I will present our things. Right, the normal story. Can everyone see my slides? Everyone's happy? All right, so thank you for everyone for the kind introduction. And yeah, today we'll be chatting about how we've streamlined the workflow for the generation of functionalized VHHs. So first, what is a VHH or a nanobody? So a human IgG has this typical structure. It's around about 160 kilodaltons. And in the 1980s, in a lab in Belgium, they discovered that camelids have an additional set of antibodies, as such named heavy chain only antibodies. If we look at our antibody, this is the area that actually binds to our antigen. So if we were to reduce an antibody to its smallest functional unit, from the camel, would be a nanobody, which is 15 kilodaltons in size. So I said it's been around since it was discovered in the 1980s. And over the years, we have a trend of an increase in publications where they're actually utilizing nanobodies. Just to note that nanobody is licensed by Sanofi and is a registered trademark. So during this presentation, I will be using nanobody, VHH, and single domain antibody interchangeably. Given the size of this nanobody in comparison to a full-sized human IgG antibody, you can see that there's a 10-fold reduction in size. This small size and just the shapes of the complementary determining regions of these nanobodies allow them to bind to conformational epitopes, which may not be accessible by these big, chunky human antibodies. In addition, we can express them in bacteria, meaning that they're cheap to produce. We can easily clone them, and so they're modular. So you can add a couple of them in a string, depending on what your applications are. So the main way of finding nanobodies is using phage display. And phage display is a process whereby the phenotype the nanobody produced is linked to the genotype, so the plasmid that is within our phage. So in our phage, we have our plasmid with our genetic information, and this is our expressed nanobody. So there were a couple of reasons why we decided to publish this protocol. So there are already several existing methods for nanobody discovery, but over the years we've added a couple of bits and pieces, and we've managed to streamline this process. So we thought that we'd share this with you. We'd also like to demonstrate the application of the toolbox of vectors that we presented in this protocol, which you can use to functionalize any nanobody, either from a nanobody that you found yourself or a nanobody from literature. And given the uptake in nanobody usage in research, we would like that this protocol be made readily accessible for any research group, hence the detailed protocol. So the aim of this webinar is there's a huge difference between reading this protocol and really actually understanding it. So it's our hope that giving this, the background information would help give a greater understanding. So when you actually read this again, you will understand why we're taking the steps that we've taken. So in our graphical abstract, we had this diagram. So in this presentation, we'll walk through the steps of how we prepare the antigen, immunize our llamas, how we create our libraries, how we use phage display to find nanobodies that bind our proteins. So from these, we then will demonstrate how we prioritize the clones that we will then take forward into different applications. So first off, we need to start with a llama immunizations. So before we do anything with llamas, we need a good ethics approval. These animals need to be looked after, fed, they need to be healthy. We don't want to cause them any undue stress. We only take as much blood as we need. So we only take 170 mils after bleeding. And a very important factor that's generally overlooked is that the antigen that you use needs to be good. If you give a rubbish antigen, don't expect to have perfect things come out. So rubbish in, rubbish out. So we immunize our llamas 3 times, and at day 66, we collect 160 mils of blood. So this is important that the antigen that we use for immunization is non-biotinylated. So here's a gel of our non-biotinylated protein. It is pretty much pure. I am happy for us to immunize this llama, our llama, with this pure protein. So once we've collected blood from this llama, we also collect serum, and we use the serum to determine, has this llama actually made an immune response to the immunized proteins? And for this, in this ELISA, we use biotinylated protein. How we make this biotinylated protein, we'll cover in a moment. So in our llama serum, we're going to have a combination of normal IgG antibodies, and around about 20% of the antibodies in the serum is heavy chain only. So in our seroconversion ELISA, we are detecting both, whereas for our actual nanobodies, we're only going to be focusing on this, but that is to come. So the layout of our seroconversion ELISA is we coat the plates with neutrabidin. So this neutrabidin has a high affinity for our biotinylated protein. And so our biotinylated protein will bind to the neutrabidin, and this will allow that all the proteins are aligned in the same orientation. So any antibodies in the llama serum that bind to our protein will then be detected by an anti-llama HRP. So this antibody will detect both the IgG as well as the heavy chain only. So if our llama has produced an immune response to our protein, we will get a green color. If not, we'll get no color. So this is a typical trace of a seroconversion ELISA. So we take serum before we immunize our llama, and as expected, there is no response. Our llama hasn't seen our protein, thus we don't expect a signal. However, with our immunized, we expect the llama to have made a response, and indeed we get a large absorbance value, indicating that our llama has seen our antigen, and there is a good likelihood that we are able to find binding nanobodies from this library. So I spoke to you about we need to prepare our proteins either in biotinylated and non-biotinylated. So we use biotin because biotin and streptavidin have a femtomolar affinity for each other, which is very important during our panning stages, which I will explain a bit more later. So there are different ways to biotinylate your protein. We are in favor of in vitro biotinylation, and this is because we can control where biotinylation occurs. So we use either an AVI-tag, which is also known as BAP, or the biotin acceptor peptide. So in our DNA encoding for our protein, we then add a C-terminal AVI-tag, and when we co-express this with our Bure enzyme and biotin, it will result in Bure adding this biotin to this AVI-tag, and we result in a C-terminally tagged single biotin protein. However, you can use chemical conjugation where biotinylation occurs at primary amines. However, a protein may have many primary amines, and it's difficult to control where it's happening or how many biotins are labeled to a single protein. Hence, we are in favor of our in vitro. So we have our blood. We collected 170 mils of blood, and now we want to isolate our PMNCs, which are polymorphonuclear cells. These cells we will use to isolate our RNA in the next steps. So how we do this is in our falcon tubes, we add histopaque, which is like a sugary syrup, and then we add our blood. It is very important that you adhere to the acceleration and deceleration settings as outlined, as this will allow us to form this gradient. And here we can see after centrifugation, we have our pelleted red blood cells, the sugary histopaque, and our PMNCs, which we need to use further on. So once we've isolated our PMNCs, the next couple of steps are just to wash away any of the sugary histopaque and any plasma. So now we're getting closer and closer to our RNA extraction step. So we have found that by using RNA ZAP in preparation of the hoods and just even on our gloves, improves our yield of our RNA for the next step. So in our next step of our library preparation, we take our freshly isolated RNA, and using a one-step reverse transcriptase PCR system, we transcribe into cDNA, so in one tube, and using these Cal1 and Cal2 primers, we are able to produce what we call VHH1. And these primers have been referenced in the Pardon paper in 2014, just for reference. And this can be used for all camelid libraries, camels, llamas, alpacas. So after this PCR, we get this predominant band over here, which corresponds to our VHH1. We will then gel extract this and use this as our template for our second PCR. So the second PCR product is called VHH2, and the primers that we use add 15 base pairs overhangs that are complementary to our vector, which will be used in future steps. So again, we run a gel and we can see that it is amplified at the correct size. So at each step in our process, you have to have a checkpoint. Rubbish in, rubbish out. Don't rush it because you're like, oh, I need to get it done. If these are not the right size, it's not worth carrying on. So to keep that in mind. So we have our insert, and now we need to have our vector. So we use a PADL23C phage vector, and it has a couple of features that I'd like to point out to you. So it has this ORI F1, which is very important for phage and viral packaging. It has ampicillin resistance. It has a phage coat protein, which is important in phage display, as well as a trypsin cleavage site, which will come into play a bit later. So downstream, when we can express protein in this vector. It has a PEL-B signal sequence, and this signal sequence is involved in sending our expressed nanobody into the periplasm, which is reducing and allows our disulfide bonds to form. And on the C-terminal end, it has a his tag and a mic tag, which can be used in affinity purifications, which will be covered a bit later. So what we do is we cut our vector, we gel extract the vector backbone from our stuffer fragment, and once again, we run a confirmatory gel to confirm, is our digested vector and our insert at the correct size? Are they sufficiently pure for us to continue? So yes, we are happy with the purity and their sizes. So now we go on to doing a test of our library preparation. So we do a small scale library preparation. So we have our vector and we have our insert. And remember the insert, we had the 15 complementary base pairs to our vector. So we then do an infusion reaction. So with this overlap, this infusion enzymes choose back some of the 3' end. They then anneal and repair and ligation occurs in E. coli. So at the end of the day, you essentially get a plasmid with your PADL with your VHH2 in it. So here we have our plasmids that we have made. We then will electroporate them into TG1 E. coli cells. And the resulting cultures, we will then do a titration to get individual colonies. So from these titrations, we can work out the library size, which is expressed as our colony forming units per mil. Another checkpoint here is we want to see, are these nanobodies that we have made in this library, are they the correct expected size? So how we do that is we pick 48 of these colonies from one of these plates. We then amplify the VHH gene, and we then run them on a piece, on an agarose gel. And out of the 48, only 2 are of the incorrect size. Excuse me. So this is a good indication that the majority of our library is the correct size and that we can proceed. So based on this library size, we know how much DNA we've put in, and that will yield a library size Y. So for us to now make a large-scale library, we want a library of around about 1 to 2 million colony-forming units per mil. So knowing that X amount of DNA produced Y-size library in our small scale, we can scale it up to work out how much DNA we need to form this library. And for safety's sake, we always just scale up, and we generally do 20 infusion reactions, which is more than enough just to improve our chances of having a really diverse library. So infusion reaction to make our plasmid. We then do electroporations, but we increase the number of electroporations as we've done a lot more DNA. And then we, instead of doing a titration, we plate these all on these really large bioassay agar plates. And this is what we use to create our library stock. So looking at this, this is the density that is equivalent to a 1 million sized library. So these plates, we scrape off the agar, and we add glycerol, and this is our master stock that we're going to use for our panning. So before we start panning, it's good to do some preparation work to save on time during your process. So we can prepare our log phase TG1s, which have no antibiotic resistance, as well as our helper phage, which has campamycin resistance. So once you've prepped them, they can be prepped way in advance or just before, stashed in the -80, and they're stable for a year or so. So before we go into panning, I need to tell you a little bit about how phage actually works. So typically, a plasmid with the REF1, like our PADL phagemid that we use, if we include this with a phage, we should be able to package DNA into viral particles, so producing more phage with our DNA of interest. However, if we use wild-type phage, the plasmid interferes with the wild-type's phage replication. So this interference decreases the production of the viral particles. So over the years, there have been a lot of development, and they have come up with a helper phage, and they're constantly working on this. So this helper phage has been designed to minimize the packaging of its own DNA and prioritize the viral packaging of the plasmid that we're putting in. So we are going to exploit that in our process. So those big bioassay plates that we've made our glycerol stocks of, so this has E. coli with our phage vectors. Now these phage vectors have our nanobody repertoire from our llama. So what we do is we add a helper phage. So remember, this is ampicillin resistance. This is kanamycin resistance. So in the presence of kanamycin, ampicillin, as well as decreasing the temperature and including this helper phage, we're going to maximize the packaging of these plasmids to produce phage that have our plasmids and that are displaying our nanobody of interest. So this is what we use for our panning. And after we've found the phage that have bound to our proteins, we need to cleave that off so that we know the genetic material of the clones that have bound. And so we want the DNA, we don't want the phage, so we put them straight into E. coli. And then the circle comes back to the beginning. So we will have enriched phage plasmids in E. coli. Right, so now that you've got a fair amount of background, we can actually go into the nitty-gritty of panning. So we do 2 sets of panning stages at lowering concentrations of protein to select for strong binders. So starting from our library that we resuspended from those big plates, so it's our library with our phage mid ampicillin resistance. We express it, we add the helper phage with kanamycin. So here we're going to prioritize the packaging of our plasmid DNA into the phage and presenting our nanobodies. And this in the morning, we get this phage, we will then precipitate it to be used in our panning strategies. So panning, we add 50 nanomolar of protein, biotinylated protein, to our phage, and we agitate it. And then we then add streptavidin beads, which will bind to our biotinylated protein, pulling our bound phages away from all the unbound phage. So we then wash away any unbound phage. And now we want to know, what is this genetic material that encoded for these nanobodies that bound? So we use trypsin. Remember, I said that there was a trypsin cleavage site here in the coat protein, so pretty much the tails. And at the end of the day, we get these phage with our plasmid. So we don't want to make any more phage, we want to make the plasmid. So we pop it into E. coli cells, and at the end of the day, we form a new sublibrary. So this is ampicillin resistant, and it is now enriched for binders against our protein. So this is the library that we then use for our second round of panning. So we grow up our sublibrary, we add our helper phage, and this is going to prioritize the making of the phage that are enriched for binding to our nanobodies, encoding for the DNA on the inside, displaying the nanobodies on the outside. So 5 nanomolar of biotinylated protein with our phage. So using a lower concentration of protein will increase competition for strongly binding nanobodies to our protein. So we wash away any unbound phage. We use trypsin to cleave that off so we can capture the genetic information that led to the bound phage, and we add it into E. coli. So this is slightly different from our first round of panning. So we've had this enriched pool, but now we want to interrogate the individual members of this enriched 5 nanomolar pan of phage. Which ones of you actually really do bind our protein? So how we do this is, once we've mixed our phage with our E. coli, we do a serial dilution, because we want to get individual colonies that we can pick and interrogate. So we pick 93 different colonies, we grow them up, and we make 2 different master plates. The first master plate has glycerol, which we use for culture. And once again, it only has our E. coli with our phage vector. While we're making this master plate with glycerol, we also make a PCR template plate with our glycerol. So important to note that A1 in our glycerol master plate is equal to A1 in our PCR template plate. So they are equivalent. And these are both utilized in different situations. So we're interrogating the individual members of our enriched pan, which one bind. For us to do so, we use our master plate with glycerol. We then culture it. So we want to grow them up, make sure the bacteria are viable again. So bacteria with our phage mid, we add our helper phage, lowering the temperature with the 2 different antibiotics, and we get a resulting 96-well plate with phage. So the phage with the genetic inside displaying the nanobody on the outside. So we will then use this in an anti-MIC ELISA. We want to see which of these phage bind our protein. So we've seen this setup before. So we coat our ELISA plates with nucleotide. We then come in with our biatylated antigen, which then orientates all the proteins in the same orientation. We then come in with the different wells, the phage from these different wells. If the phage binds, it will be detected by an anti-M13 phage secondary antibody. If the phage doesn't bind, we will get no signal. So this is a representative of one of these anti-M13s that we've run previously, and we can see that a couple of them are indeed binders, whereas the majority aren't. So we will then focus. We want to know, well, so these phage with displaying nanobodies, these bind. What is the genetic material that encodes for these binders? So I remind you where this is. So these are our hits. Remember, we got these hits from growing from a master plate with glycerol. So for us, we want to now work out the genetic material. So we use our PCR template plate. Remember, A1 here is equal to A1 over here. So we will then pick each one of these wells from our PCR template plate. We will then amplify the VHH region and then sequence it and submit any of these sequencing results into IMGT. IMGT is a website that allows us to demarcate where the framework regions are, where the complementary determining regions are, as well as a host of different functions. But for us, that's pretty much what we're looking for. So the next step is, how do we analyze the sequence data? So I'm going to give you an example of sequence of anti-GFP nanobodies that I've received from literature as an example of how we would sort. So we predominantly sort based on our CDR3. So the CDR3 is the main determinant of binding affinity. There are other factors, but this is our main sorting tool. So if we were to sort by the identities of our CDR3s, we can see there are 3 clusters: this light pink cluster, this dark pink cluster, and one little singlet. So how do you know which one binds best? Or does it bind at all? So remember, this ELISA was a single well. Is this real or not real? So what we do further on is we're going to do a small scale expression. However, there have been different times where we've had 20 different clusters. Are you going to now do a large scale expression of 20 of them and taking the chance that half of them may not actually be indeed binders? That takes a lot of time and effort. So we introduced a small scale expression. So we take a representative from each one of these clusters, and we express this. We want to see, are these nanobodies expressible? Can we purify them? And do they bind? And if they do bind, how tightly do they bind? So this is so what we use is, once again, we go to our master plate. We know where the locations are. We then will express it and induce protein expression using IPTG. So remember, this has a PEL-B leader sequence, and it's going to be directed to the periplasm. So after overnight expressing with IPTG, we will then isolate the periplasm. And using that periplasm, we will then utilize the his tag on the C terminus of our PADL vector, and we can then purify using nickel affinity chromatography. So our example of our nanobodies against NIFG, we can see that these both 3 expressed really well and they purified really well. So now, do they actually bind the protein? So we use a titration ELISA to validate this. And this is what a layout of a well. Once again, Nutravidin to orientate our biotinylated protein in the same orientation. We will then add our purified nanobody from here. And if this nanobody indeed binds, it will be detected by an anti-VHH secondary antibody. If it binds, we'll turn green. If it doesn't, it doesn't. So we do a number of concentrations of nanobody. And if we have a look in blue, this A8 is the strongest binder, as it has the highest signal over the largest range of nanobody concentrations. Whereas B7, it's already a low absorbance to start with, and it doesn't change much as the concentration decreases. So this generally is sufficient for most labs. So some labs may have surface plasmon resonance or biolayer interferometry. So these are all label-free ways of working out your protein, your binding interactions, KD or off rate. So it works very similar to an ELISA. So on your sensors or your chips, you will load your protein, like here. You will then add your nanobody, so purified nanobody, and you will see association. It's binding, and then we measure how quickly it falls off, so our dissociation. So a representative image is for our NIVG nanobodies. We can see that all 3 nanobodies associate, and they dissociate. So we can work out, we can measure the rate of dissociation, so the off rate, and the off rate is concentration independent. But for our case, there was only 3 of them, so it would be easy enough to upscale this. However, if you had 20, you could use this off rate to prioritize which nanobodies to pursue for large scale, depending on your application. So we've spoken about how we found these nanobodies, how we can prove that they bind. So we have a nanobody, now what do we do? So another thing that we've done in our paper is we have published our vector toolboxes. We have a toolbox for E. coli, and the beauty of these toolboxes is that each set utilizes the same restriction enzymes and all have the same forward primer, and they have slightly different reverse primers depending on their tags. So in this vector, the E. coli vector toolbox, we have a vector for his tagged only, so for crystallization, for epitope detection, for biotinylation, and for conjugation, either by chemical or enzymatic conjugation. So in addition to our E. coli plasmids, we have 2 sets of mammalian plasmids. The first one is to add a FC domain. Once again, same set of restriction enzymes, same set of primers, and the exact same for if we're going to express our proteins as fusion proteins, so in frame with GFP, YFP, or M-cherry. So I'm going to explain a couple of how we actually do this subcloning. So using any of the nanobodies that we found in one of our screening processes. So we have our nanobody in our phage vector. We will subclone into pop and VHHs. So for example, for crystallography. The way we do this is very similar to how we prepared our library. We can also clone them into the BAP vector for biotin pull downs, or we could express them in the pop and cis or the EFP M-cherry for visualizations. So to quickly go through how we actually perform this cloning, we will then use our nanobody, our plasmid from in PADL vector. We will then using those primers as detailed that have the 15 base pair overhangs that are complementary to our vector, amplify our nanobody, insert it into our cut vector using infusion to create a new vector. This vector we can then either put into E. coli, or if it was mammalian, do transfection. So the same process is applied through all the different vectors that I'm going to describe now. So most importantly, and more interestingly for us, is we want to demonstrate that we can use any nanobody, whether it be from a nanobody that we found or a nanobody in literature, to put into these vectors, for an imaging application. So we're going to use antivimentin. So these detect filaments in HeLa cell or just in cells. And we got the sequence from literature. We subcloned into our POPIN VHHs, cis, and it was our intent to use this for in vivo visualization. So if we look at this vector, it has a single C-terminal cis residue, and this cis has a thiol group, and this makes it reactive to maleimide. Important when we're doing large-scale expression of this is to use this in the presence of TSEP, which is important to reduce disulfide bonds, and this frees the C-terminal cis ready for conjugation. So we expressed our antivimentin nanobody in this vector with the free cis, and then we added a maleimide conjugated alexaflor 647. There's lots of words in there. So to confirm that coupling occurred, we could run the sample on a gel, and we could see that there's fluorescence for our nanobody that we've coupled at the same place where the Kamasi is seeing it. In addition, we used UV spectroscopy, and we could see that there's a clear difference between unlabeled vimentin nanobody and labeled vimentin nanobody. So alexaflor 647 is towards the red end of the spectrum, so we're expecting a peak between 600 and 700. So we've made this nanobody, now what? So we added this fluorescent nanobody to HeLa cells, and then we prepared slides, and we used confocal microscopy to show that our vimentin conjugated to this fluorophore was indeed able to detect the vimentin filaments in HeLa cells. So what about live imaging? Well, using the same antivimentin sequence, using the same method of subcloning, we can subclone them into POPIN-E either in frame with GFP, YFP, or M-cherry. So how we go about that, we have our vector that we've now made with our vimentin and our GFP, and within the actual HeLa cell itself, it will express our antivimentin nanobody with GFP fusion protein or with YFP. So using live cell imaging, we can see that both the GFP and the YFP are able to label the filaments within these HeLa cells. So in summary, we've spoken about our immunizations. It's important for you to have animal ethics involved. It's important that your nanobody, your protein is good. Rubbish in, rubbish out. It's important to do a seroconversion ELISA to give you an idea that, yes, this will work or this may not work. How can we adjust things? We then spoke about our library preparation. It's important to use RNA ZAP when you are isolating your RNA. We spoke about how we prepared our libraries. Once again, checking our insert and our vectors, how we scale up our library so that we can get a big enough and diverse enough library for us to actually do our panning display. We spoke about how helperphage helps with panning and how by reducing the protein concentration that we use with panning, we can improve the binding, the isolation of strong binding antibodies, nanobodies. So once we have them, we will interrogate the individual colonies to see which ones bind our proteins. We sequence them, sort them by our CDR3, and using that, we are able to prioritize which nanobodies we can actually go for large-scale expression. Having shown that the whole process works, we showed that using either a nanobody from our own work or a nanobody from literature, we can subclone them into any one of our vector toolboxes that we have presented, and we can use them for imaging applications. And this is just one such example. So you may ask, okay, well, I can't make a nanobody. Where do I find these nanobodies? Well, there's no point if there's a nanobody, for example, against GFP, there's no point in you going to try and do phage display to find your own GFP nanobody. So there are a couple of nanobody databases out there, and we'd like to highlight the single domain antibody database, as well as the natural antibody database. So these two have an automatic scouring of public resources such as patents, PDB, GenBank, and NCBI to find sequences and associated proteins. And recently, the Nanosaurus was published. it is their hope that they're trying to kind of be like a PDB. So when you publish a paper, you will put all the relevant information, how this nanobody was found, what library did you use, what kind of panning did you use, so that you have more of an idea of the background story. So all this work is not done by myself. We have a large team, as detailed here in our good fun photos, but the most importantly is the llamas. Without them, we actually can't do very much. So yeah, that's it for me. Thank you very much for your attention, and I'm happy to take any questions.
Thank you very much, Laura. That's really good. Yeah, so there are a few questions popping up in the chat. So perhaps we should pick some of those up as we go now. This question came on quite at the beginning in relation to the seroconversion test.
Yes.
Very specifically asking why we don't use an IgG2A specific reveal, as it were. I guess this is bearing in mind that most of the heavy chain only antibody isotype is IgG2A in the llama. So
okay,
that's
okay.
Comment on that.
Sure. So we were starting with, so when we first started with the seroconversions, we were using both the polyclonal anti-llama as well as just the anti-VHH. So we were hoping that they would both show us the same signal. So bear in mind that the VHH is only, the heavy chain only constitutes 20% of the immune response, maybe sometimes less, depends on the llama. So we were having weird results when we were using the anti-VHH. Instead of having a nice titration, it would do weird little triangles. So we thought, instead of muddying the waters, let us go with using the polyclonal anti-llama. And we've then started setting somewhat thresholds on what a good response is and what isn't. So yeah, I hope that answered the question.
Yeah, and probably just to elaborate a little bit, I think we haven't yet seen where we've had seroconversion using a polyclonal anti-llama antibody that we'd never have then gone on to find a VHH to that antigen. So that's a good point. We could think about making more isotype specifics and maybe homing in, but I think we have had issues going straight to the VHH as Lauren just explained.
Yeah.
A few questions about the cloning strategy. I tried to answer one, I think, more generally, but comment on infusion cloning versus the conventional T4 ligase cloning or methods in general. I mean,
yeah,
your experience, Lauren.
You can. They both work coming from a T4 ligase background. The infusion is just so quick. It's all about streamlining. So if people want to carry on using T4, go for it. But our experience, the infusion cloning works pretty much all the time. Your plates are predominantly white. You're lucky to see one blue. If you see one blue, you get really excited. You're like, you know that you've put XGAL and IPTG on your plates.
Okay, thank you. And perhaps now thinking a little bit more about the strategy for panning or screening the VHH library. A couple of questions around this. One is, how variable are the sequences that you recover from the panning process? You know, do you get all the same sequence or do you get variety? And related to that, I guess, is why do just two rounds of panning?
So first question, it all depends on the protein, depending on how many different sequences you get. We have had entire 96 well plates where the entire plate was green and we had 2 sequences. We have had some where there's lots of clusters, or we have had ones where there's only singlets. It's all very protein dependent. You've just kind of have to roll with it. And that's why I think adding the small scale, just have a look, have a glance in and see, is this going in the right direction? Second question was, how many rounds of panning? You could do many. I've seen people do 6, 8. It also depends on if you're doing yeast or ribosomes. For us, 2 is sufficient. 2 can get you done with, you can do 2 rounds of panning in a week and you can get a yes, no answer. The seroconversion should guide you. If you have a strong seroconversion, that should guide you that 2 rounds of panning should be good enough. This also may lead into the question of versus naive versus immunized. Immunized, you're expecting an immune response, you're expecting a high affinity binder, so 2 should be sufficient. However, if you use a naive library, you may need to increase the protein concentration, or you may just do 1 round of panning and have a look-see. So do your anti-M13 to see, have you got binders? Because using a naive library, you haven't had any affinity maturation of your protein. So it all depends. So for us, this 2-step is a good starting point, and then you have to wiggle around and see what you get.
And I guess it's also worth making the point you can manipulate the concentration of the antigen to force to more stronger interactions. So
exactly.
The 2 rounds are not done in the same concentration.
Yeah.
Slightly related to that, very specific question about the temperature of the incubation part in the literature. You can do these panning reactions at 30 rather than 25.
Yeah.
Is that
You can do 30 or 25. For us, we do our protein expression at 25, and it was just like, it just kind of happened, and it works just fine. It's ease of use, but 30 works just fine.
Because it's convenient to do them at lab temperature.
Exactly. Yeah. So that at least I would rather keep it at 25, where the majority of our other things are doing, so that you're not getting changes. Like, oh, 1 week you're like, oh, I remember it was supposed to be at 30. On the next week, oh, you forgot that it was supposed to be at 30. And now you're adding variation. So keeping consistent at 25, that just worked for us.
Some more questions coming in. There's one just general question about the timeline. Perhaps you could just...
Yes.
How long does it take from immunization through to having an antibody in your hand?
Well, if everything works to plan, nothing goes wrong. The seroconversion is perfect. Your protein is perfect. Everything is perfect. 3 months. But that's if everything is perfect. But that's not science.
And I guess most of the time is the animal. The immunization is like a
exactly.
12-week. Yeah. So it's not a quick thing, and I think Lauren's already mentioned that we do also screen the libraries that we have, sort of naive to the antigens, as a way of looking for things that may be already there. In that context, talking about the library and stuff, question here about the sequence of the CDR3. How diverse do they have to be to not be clustered? Do we have a criteria how many changes, or how similar do they have to be?
So once again, it depends. Like if you have conserved changes, so like an alanine to a valine, you could consider them at the same. If there's a whole lot of different conserved changes, you could maybe decide that that's a separate cluster. So we are working on different ways of, so before we were just, CDR3, just cluster them, that looks fine. So we're starting to see some of our projects, some of the sequences are really closely related. So we've started doing alignments and phylogenetic trees, just to kind of get us an idea of, do they cluster separately, or is this a completely different node? There's other work going on, but that's as much as I can do. Have a look, and once again, you're doing a small scale. You can do a large number of them. So if you've got extra space, why not just include one if you're unsure if they're slightly similar or slightly different? They may have a difference in antibody affinity, nanobody affinity, sorry. So yeah.
And sort of bearing in mind that you've got your binders, question here about whether there's any limitation to the type of epitopes that nanobodies can bind to.
So nanobodies bind to conformational epitopes. So nanobodies are more, they're loops, and so especially the CDR3 is quite long. So that way they're not really going to sit to a flat something. They want to get into the nooks and crannies that big chunky IgG molecules can't get to.
So I think that the consequence of that is that there's very few, if any, nanobodies out there to just strictly linear epitopes.
Yeah.
Where obviously you can generate conventional antibodies to those.
Yeah.
I think I've come to the end of. Well, there's a very specific question here about the Calo 001 primer, which perhaps I should answer because I think it's just to clarify, that is, it doesn't bind in the variable domain. It's basically at the CH2 VHH junction, so that's why it's nested. The secondary primer that, as Martin explained, is nested to that. So it's basically to a conserved constant region.
So if Anton wanted to know a little bit more, go and see the Pardon 2014 paper in the supplementary figures. They've got a really nice diagrammatic diagram illustration to show where it actually binds in the different classes of the llamas or camelid antibodies.
Question, the specificity or scope of antibodies for partially or degrading proteins, you know, for detection in forensics and such like that. I guess it's again a question of the scope. How what's the diversity of nanobodies that we're aware of?
You could potentially make a nanobody against anything. It all depends. Like, I showed in the PubMed graph, it's only started to tick up recently and predominantly dominated by COVID. Due to the pandemic. So people are hopefully starting to catch on to this and starting to do so. And everyone's got a different protein. So the sky's the limit. If you're interested in that, well, has anyone done this before? Is anything similar? Yeah, you could.
And I think, again, just to emphasize what Lauren concluded with, there is these resources are now being developed to really try and make nanobodies or sequences of nanobodies that are out there already in the literature. I think one of the big challenges for everybody in the field is the nomenclature and the fact that nanobodies have got a very idiosyncratic naming convention. So part of the Nanosaurus initiative, as Lauren explained, is trying to do something like the PDB and have defined identifiers that we can all work with at least once they're published. And then, because I'm sure the different antibodies probably have the same name, and there can be a lot of confusion in the field, isn't there? Phosphorylated site, then antibodies to phosphorylated sites. Yeah, we're interested to try and do that actually. So we could let you know if that's successful. But it would probably be in the context of a domain rather than trying to do it with a phosphorylated peptide. Obviously, the monoclonal or polyclonals that will pick out phosphorylated sites in peptide sequences. I don't think nanobodies would work in that way. I think I'll try. It's any new questions. Thank you for the audience for the very nice comments. I hope that Lauren can see those in the chat as well. Very much appreciated, and I obviously appreciate people joining us for the webinar today. Perhaps I'll hand you back to colleagues at Bioprotocol. I think we're closing on the hour, so it's probably a good time to conclude. So I'll hand you back to them. Thanks again, Lauren.
Thank you.
Thanks, Ray.
Thank you, Dr. Eisen, Dr. Owens, and thank you very much for the very interactive chat session. There has been a lot of questions, and it was a great, insightful webinar. Thank you very much. And yes, as I mentioned earlier, this webinar is being recorded, so you can go back to the recording anytime later and brush up your knowledge on nanobodies. And yeah,